I'll do the straight version of this first and tell you a bit about Craig Venner you probably already know. He is the founder and chairman of the J. Craig Venner Institute. He got his bachelor's and PhD degrees here in town at UCSD. He developed later on an idea and a discovery called expressed sequence tags, ESTs, which allowed for the first time a new technique for doing genome studies, which he developed, which he calls shotgun decoding. And in '98, he founded a company which you probably all heard of called Celera Genomics. And the very tactful way of saying this, I think, is that as he says himself, in February 2001, in Science, he was one of the publishers of the Human Genome in the journal Science. We'll get back to that in a minute. He has received the 2001 Paul Ehrlich and Ludwig Darmstädter Prize and the 2002 Gairdner Foundation International Award. Those of you who track this stuff know that Craig is the guy who challenged the United States government on its 58-year program to do the human genome. That's a joke. And condensed that time to relatively few months, basically, and has since decoded any number of mice, flies, scientifically interesting animals, and bacteria using this technique, which is just radically faster than anything else. I think that Craig is interesting personally because he's unafraid to do something different. He goes beyond the definition of thinking outside the box. He finds a very direct way from A to B that nobody else seems to be able to think of, and he seems to be courageous enough to do it, regardless of what they all may say. And we all benefit when that happens. So I think that's probably enough. I'll also mention one more thing. Tomorrow afternoon, with Larry Smart, we'll have a chance to go over to Larry's CALIT-2 project, as we did last year. And Craig and Larry are now working together on a thing called CAMERA, which, excuse me for doing this is really difficult. And I'm told this is Larry's fault. CAMERA stands for Cyber Infrastructure for Advanced Marine Microbial Ecology Research and Analysis. And Craig will explain it. But basically, we're going to have a chance to look at some of the output from that study and be some of the first people to see five new things that Larry has arranged for us, one of which is this partnership between Craig and his institute, CALIT-2, Scripps Institute, the Gordon and Betty Moore Foundation, the Center for Earth Observations and Applications at UCSD, and the supercomputing operation here at UCSD. So an amazing amalgam, and Lambda Rail. So all these different groups getting together to create what Larry's calling collaboratories, where people can actually come in virtually and see data, work on data, use these tools, which are 100 gigabit glass tools. Really amazing. I think we're very lucky, having chosen San Diego, to have the ability to go to what Larry more and more often calls the FiRe lab and see this amazing stuff. So I hope you'll all take advantage of seeing that. And Craig, thank you for joining us.
Well, thank you for the extremely kind introduction and the invitation to come here and actually allowing me to break your cardinal rule about PowerPoints. But see, my argument was I was trying to explain some complex biology to a bunch of non-biologists, and we need some visualization techniques. You'll see some much more dramatic ones, as you heard from your visit to Larry's operation tomorrow, and we're limited to simply 2 dimensions today. But I want to take you through a brief history of genomics, which is a very young field that many people feel only started in 1995 when we sequenced the first genome. But most people view genomics as this technical field that because we had better machines and better computers, that's all there was to it. And in fact, it was as much philosophy and strategy as anything else, and that's why there was a 15-year, $5 billion government program that also involved all the European Union and China and Japan and other countries, that they thought it was such a massive problem that it could not be solved in a simple fashion. But by adopting concepts such as massively parallelism, for those in computing know that that's a very simple concept that's been applied very nicely. But biologists are a little bit more simplistic, I guess, and they wanted one machine to sequence the human genome. And when various automated devices came out, the argument was they won't work because they didn't have the throughput. When we wanted to double our capacity, I bought a second machine. And that seemed to be a radical concept in NIH and biology. We took that up to 300 of these $350,000 machines, and it had a nice impact. Randomness is maybe a more important concept, and it's a thread that's gone through everything. I was actually told when I started my graduate work in UCSD in the early 1970s that it was going to be impossible, nearly impossible, to come up with anything new in biology because it was all known. And now through the concept of randomness, anybody can randomly take a cup of seawater and make more discoveries of new species than were made in the last decade, or just randomly picking one of the 6 million new genes we just put into the camera database and study those and make amazing discoveries. And you'll see this thread through things. So this started with just searching for human genes. I spent the first decade of my career trying to isolate the adrenaline receptor, being fascinated with adrenaline. And it took a decade to finally get enough protein to get a little bit of sequence to clone the gene. And one day, just flying back from giving lectures in Japan, I read my book on the way over. I had nothing to do for 12 hours, so I thought of a new idea of, instead of trying all this work to clone genes after a decade of work, why not just randomly select some from a cDNA library? And that led to the Est method. And this was the first paper in 1991, and it described 337 new genes, which seemed really radical at the time, given that genes were discovered on a once per decade, per career kind of phenomenon. This number now seems remarkably small, given the fact that we just published a paper with 6 million new genes in it. And it shows how things could scale up. We generated more and more of these sequences, and we had to develop a new algorithm to assemble these. And it turns out the biggest breakthrough in genomics was new mathematical algorithms, not a new machine or a new computer. Obviously, those things help, but the algorithm for assembling these sequences, simple pairwise comparisons, is what gave us the notion of just shattering the genetic code into little pieces and making libraries of the DNA. And this is where they have to be made perfectly, because if you're depending on randomness and random selection, you have to have truly a spectrum of every piece of DNA there. And you just randomly select those, and you can calculate roughly how many you need. And for the first genome, we took 25,000 pieces and sequenced them. And you can see it's relatively simple when it's the number of sequences squared. So it was roughly 10 to the 8th pairwise comparisons, and that wasn't a big computing triumph, but it was a biological triumph because it allowed us, after 4 months, just to computationally reassemble the genome in the computer. It's not that we're so bright, it's just that other people are really dumb. Because the way this was being approached in the government was to take 10 years and 1,000 scientists to do the same experiment by breaking it down to little pieces and sending one to each of hundreds of labs around the world and having them sequence the little piece and of varying quality data, and then trying to get together and put it all together. And so the yeast genome literally took 1,000 scientists 10 years to do. It's now an experiment we can do in 1 day or less, and they're not even good graduate student projects anymore. We tried to get money from the government to do this experiment, and nobody believed it would work, so we used our own money to fund it. But the government in this country, and I think the European Union certainly not any different, won't fund new ideas, but once an idea is established, they'll fund it. And once we published the first genome, we got more money than we ever wanted to sequence every genome under the sun, and we did most major human pathogens and tried to scale it, but the human was still the major goal. The big breakthrough came when we developed with a team headed by Gene Myers and Granger Sutton a whole new set of algorithms. This team of mathematicians and computer scientists wrote about 500,000 lines of computer code in about 3 months for these new algorithms to try and assemble much larger genomes. So the fruit fly genome is 180 million base pairs, where the first genome was only 1.8 million. And to cover that, we needed to do roughly 3 million sequences. So now you can see it's that number squared, so all of a sudden you end up now with 10 to the 12 pairwise comparisons. And it took a lot more compute capacity, and particularly a lot of computer memory to do this with the early algorithms. So in 1999, we built the third largest computer using alpha chips, and it was only 1.5 teraops. Today, that wouldn't even rate in the top 500. We have a computer that's almost 3 times the size and is the 125th largest or something. But fortunately, the scale in this genome was equally important to the first one because it showed we could work at basically any scale. And after the fruit fly, human was just very straightforward to do. And we sequenced the first set of human genomes was actually from 5 people, 3 women and 2 men. I was one of the men that it was compiled from. And it took only 9 months to do this, not 15 years. And it was about $100 million, not $5 billion as was spent on the taxpayer side. But here we did 27 million sequences. And so now we're up to 10 to the 14th pairwise comparisons. And we thought this was really a big compute until we got into the environment. And I think people have seen examples of this data. This is a piece of what graphically a small section of your genomes look like, or at least my genome does. And you can see it's those little, it looks like a musical score, but those little notes are actually genes. And you can see they're very heterogeneous in their distribution. Some areas are extremely dense, and other areas, we call them deserts, where you can go for millions of base pairs and not find any genes at all. We have to very quickly get into this era of individualized genomes, and that's going to start in the next month or so on a scale that's important, but 1 or 2 doesn't matter. We need on the order of tens of millions of genomes. And so now a complete human genome, I'll show you, is on the order of 6 billion base pairs. And it's like what we're doing with the environmental data. The sequence is only the small part of things. We need all the corresponding clinical and phenotypic information on everybody associated with this. So the compute challenge for doing a multivariate analysis on 10 million human genomes with all the clinical outcomes is a challenge that I don't think anybody's truly ready for. But as soon as we do that experiment, we will be able to answer fundamentally any question about nature versus nurture. And we'll be able to know the hundreds of gene changes that contribute to human traits and disease, where right now the thinking is extremely simplistic of people trying to measure single base pair changes and trying to attribute that to specific diseases. So over the last 4 years, we've been adding a lot of sequence data to what we initially generated, and now we have what we're calling the first diploid human genome, where each of us get 2 sets of chromosomes, one from each of our parents, And it turns out those two sets of chromosomes are more remarkably different from each other than anybody imagined. So this notion that everybody's been going on in genetics, we differ from each other in 1 out of 1,000 base pairs, is a tremendous underestimation of human-to-human variation. And I think that will start to show up soon. There's these other changes called indels. They're insertion, deletion mutations. And we actually have more base pairs in indels than we do in these single nucleotide polymorphisms. And these have a huge impact when they occur in genes. This is one where it changes the number of DNA binding domains on a zinc finger that controls expression of other genes. The environment is going to turn out to be at least equally important to the genetic code. Most geneticists don't want to believe that right now because we can measure the genetic code. We don't know how to measure the environment. And so we've tried to use some of the genetic tools to measure the environment and understand the scope of what's there in the environment. As you heard in the earlier comments, can be the millions of microbes living in your intestine, in your gums. We're doing an air genome project measuring the number of microbes in the air we're breathing. And roughly each liter of air in this room is going to have on the order of 10 bacteria and maybe 100 viruses in it. So you're actually exchanging DNA with your neighbors without even doing it intentionally right now. So, you know, we're actually living in a continuous milieu of biology, microorganisms, and DNA. When we go out to the ocean, right out here, each milliliter of seawater has about 1 million bacteria and on the order of 10 million viruses. So those of you who are going to go swim in the ocean tomorrow, if you swallow a mouthful of seawater, you can do the simple calculation of how many different organisms you just consumed. You know, if you look at television and TV ads, you know, we can all be very frightened about these microorganisms, but in fact, for some reason, they don't kill us. Very few organisms are, in fact, human pathogens. And most of these organisms worry more about killing off each other and competing with each other than they do humans. And they provide unique sources of all kinds of things, such as antibiotics. Now, after sequencing the human genome, we decided to take these same tools and just apply them to the environment. So we just decided to go out in the ocean and take some samples of water, filter it, isolate all the DNA, and shotgun sequence the environment. And we did this in the Sargasso Sea, which was supposed to be a desert with very little life in it. And we discovered as many as 40,000 new species in a barrel of seawater. And people could not understand how all that diversity occurred. And so we started the Sorcerer II expedition. Just last month, we published the first results of this between Halifax and the Galapagos in the special issue of the Public Access Journal of Biology, describing first just the expedition and the sampling and what it meant. So this was our route, and the blue dots just show the samples of what's described in this paper that described on the order of 6 million new genes. We're now fairly close to complete with sequencing all these other samples from around the globe, and we'll be doing an analysis of the whole globe. This is our research vessel, the Sorcerer II. It's a 95-foot sloop, and it really angers my colleagues that I go out sailing and have fun doing this. Those in the Department of Energy complain they get sent to acid mines, and I go out sailing around the equator. But, you know, they're in the Department of Energy. Now, the protocols are remarkably simple, and I think most of the concepts that we've dealt with were simple concepts, just tools that almost anybody could use. We take seawater and we filter it through a series of filters. First a 3 micron filter, then a 0.8, then a 0.1 micron filter. Everything that goes through that's going to be the size of a virus, and we collect those with a much smaller molecular weight cutoff. So the first 2 filters have mostly eukaryotes on them, single-cell organisms, and the 0.1 is mostly bacteria and archaea, and what goes through that are mostly viruses. And it's very simple. After you do the filtration, you just put this thin filter paper in the freezer. When we'd get to a port, we'd put them in FedEx and send them back to the lab, where we can do a lot of sequencing. So with these machines, we could do on the order of a 250,000 sequences a day. Each sequence gives us roughly 800 letters of genetic code. So you can see it adds up to a lot of data very quickly. Here's a blow-up of the regions we looked at in these initial samples. They contained open ocean, Gulf Stream, estuaries, et cetera. And this is a rough geographic map. A political map looks very different, and that was a key part of these experiments that I was not aware of until we went out to do them. For example, the large Caribbean Sea here, there's not any international waters. Actually, there's a tiny, roughly a square mile right about here. The rest is claimed by 1 or more countries. And after we published the data of how many genes we could find in the Sargasso Sea in a barrel of seawater, all these countries decided that they definitely wanted to own all the water and wanted to restrict the science that was done in those. So it became a tremendous negotiation through the State Department to be able to take a barrel of seawater and filter it to ask some scientific questions about it. And you can see there's some blanks here where we did not take samples, and some of this water is claimed by 2 or more countries, and they patrol it with gunboats to make sure you can't sample their water. So the concept of science and what's free and what's not on the planet is certainly changing dynamically. This is slightly a different view, and we wanted to ask simple questions about the degree of diversity, where differences between warm and cold water. The red or the dark is the Gulf Stream, which originates down here between Cuba and Mexico, comes up and goes across to keep England and much of Europe out of the ice ages. There's a big danger of the Gulf Stream being cut off or diverted, and it's happened before in history, apparently during the ice ages, and it's one of the consequences that people are very concerned about with climate change that we have to take seriously. But there was a huge difference between what was in the warm water and what was in the cold water. In fact, now we can identify where a water sample came from in the world by the genetic code that we find in that sample. In fact, 85% of the sequences and species are unique every 200 miles. So instead of the ocean being this giant homogeneous soup, it's actually millions and millions of microenvironments dynamically changing, but what's in each of these regions is pretty unique. So how does all this diversity exist? How can it live if there's no nutrients, for example, in the Sargasso Sea? And I think the most interesting discovery we found is basically each of these microorganisms have a photoreceptor in it, very similar to the molecules in our eyes that allow us to see. These photoreceptors are restricted to see one wavelength of light. So it's like each bacteria having one eyeball that can only see blue light or green light. And we knew very few of these. In fact, there were only 2 microbial ones that were known before we did this experiment. So all the red were what was just in the initial Sargasso Sea experiment. But we've continued to discover these and trying to line up a couple thousand of proteins to compare them is taking unique graphical and informatic tools. And some of the gene families we have now, we have over 35,000 members. So trying to line up 35,000 proteins to compare their structures is a new computing challenge. But there's good reasons to do this. Just looking at a single amino acid residue can tell us the wavelength of light that these receptors see. A single change in a single letter of the genetic code will change that amino acid. And so we asked simple questions going back to the geography of, did we see any geographic restrictions? And we're absolutely surprised to find that in the different environments, the receptors and the organisms that survived saw unique wavelengths of light. So those of you who've ever sailed out in the Sargasso Sea know that it's a deep indigo blue. So the organisms that are selected to survive there see primarily blue light. You get into coastal waters where there's a lot of algae and chlorophyll, they see primarily green light. In fresh water, such as in the Panama Canal, they see over this entirely green light. We found a number of new derivatives, and we don't know the wavelength of light they see, but the possibilities are that we'll see things everywhere from infrared to ultraviolet, even though we can't see in that range ourselves. But this gives a different view of evolution in the sense that a single base change in the genetic code, in this case, can determine the survival of the organisms in each of these environments. A study recently published by a Swedish group, in fact, proved that these photoreceptors deriving energy direct from sunlight drive and provide the energy for growth in these cells. This is not photosynthesis. This is totally a different mechanism. This is a major mechanism for carbon fixation in the oceans that until our studies was completely unknown and has a huge impact on understanding the carbon cycles in the ocean. Now, when we look at these sequences, we had to come up with new graphic tools, and I think you'll see some of these tomorrow at Cal IT Squared, where, for example, trying to look, each one of these little bars represents 900 base pairs of genetic code, and we can spread out a sequence of a sequenced genome and compare all the sequences in the environment to that genome. SAR11 was supposed to be a single organism that existed in the Sargasso Sea, and a large percentage of the data we found matches that in some degree, but you can see it varies tremendously between 55 to 60% up to 100%. Now, we're within 10% variation of all mammals. So some people have suggested more than 10% would be a definition of a new species, but that would make us roughly the same species as mice and rats and giraffes, so we don't like those definitions very much. But here's divergence going back 40 or 50%, meaning these organisms have diverged from each other maybe 10 to 100 million years ago, yet they all coexist with similar gene order in their genomes, similar gene contents, but variants that all seem to coexist. And this goes against what many people thought about evolution, that there would be a single surviving organism in each environment. So somehow we don't understand this coexistence. We can try and understand the diversity by taking any slice of this data and making a phylogenetic tree. And this becomes pretty interesting. They're color-coded by site, and you can get exact geographic coordinates off of the camera database. For example, the middle graph can look at samples that are from the Atlantic versus the Pacific, and there's no clear-cut pattern. The bottom is the most fascinating one to me. The switch between blue light and green light happened at least 4 times back and forth in evolution. So it wasn't a one-way switch. It's sufficient evolutionary pressure to happen numerous times. And so basically you can add any parameter that you want to study to any of these slices. Some of these graphs we see huge gaps. It turns out these are where there have been viral insertions in a particular genome, and we can start to build and reconstruct the various viruses, and then go back because the organisms we had were infected with viruses in many cases. We can study the ratio of viruses to organisms. We found these varied as much as 20-fold. Most people think that the density of these organisms in the ocean are controlled by the viral infections. One way, perhaps, to greatly increase carbon cycling and CO2 fixation could be right here in the answer of the viral infectivity and why it varies by as much as 20-fold and affecting the concentration of organisms. Lots and lots of questions came up with this data. For example, how much real novelty was there with it? Was it like the photoreceptors? There were just a lot of variance around what we already knew, just distant relatives of known families. And if we're discovering new families, what is the rate of discovery in it? Is it like what people thought in the late 60s and early 70s, that we in fact know most of biology? Well, this took a lot of computing. It took over 1 million CPU hours, but we took all the data we could get from any public data source and assembled it together with our ocean sampling data. It turns out our database from the ocean was roughly twice as much as existed from all prior molecular biology and biochemistry. But to do pairwise comparisons on this took over 1 million CPU hours to put it together. And here's what we came up with, roughly 40 to 50,000 major gene families. But any sample from the environment, we could go right off here off the beach, take a sample and sequence it, and we would discover new gene families in a linear fashion. The public databases that people thought were the measure are in fact a subset of all of our data. There were 115 gene families in the public databases that we have not hit yet, but we came up with close to 4,000 new major gene families that people had never seen before, and the discovery is going on in a linear fashion. This is just to remind me and to remind you that we're on a small portion of this species tree. We're up in the animals. And so if we look at mammalian gene discovery versus microbial, the mammalian gene discovery is basically saturated. So doing the sixth groundhog or another giraffe is not going to get us any new mammalian genes. It'll give us variance in terms of genome content of what constitutes a groundhog or a giraffe. But in the environment with microbial systems, we're still very much in the linear phase of discovery. To try and put some of this data in context, Gordon and Betty Moore Foundation gave us another grant to look at 160 genomes from species that scientists had isolated around the world to try and help with this. We call this the Moore 160. So instead of 10 years to do a genome, this was a side project over the last year to sequence 160 genomes, and they're being added on to this database, trying to put the data in context. So the data is growing exponentially, and you can see on the right-hand side, some of our approaches have added to this growth quite substantially. But we've just doubled overnight this entire data set, and we decided we needed a new structure to try and deal with it, because it's not just sequence data, it's all the associated information we want with it. So in collaboration with Larry Smarr, we got a $24.5 million grant from Gordon and Betty Moore to form the camera database that you'll see a lot more actively tomorrow. But it's unique in the sense that it's not just a data collection of sequences, it's a massively parallel compute infrastructure that scientists can apply from anywhere in the world via the internet to do computes on that they're very unlikely to be able to do in their own institution. And it has all the metadata associated with it, the geographic locations, the chemistry that went with each site, satellite imagery. So one satellite image can probably take more data storage than the basic sequence data itself. And this is a great paradigm for what we're trying to do with human. So the human genome sequence that we have of 6 billion base pairs is nothing compared to the imagery from one CAT scan, if we're going to store all that information and have it be compared. So where does all this take us? We certainly know that the population of our planet is still continuing to grow, but these numbers have been revised, all due to a single virus, the HIV virus. These numbers have been revised down from 12 billion to 9 billion people. But these 9 billion, as we're adding the next 3 billion on, are consuming more and more resources. And these numbers are staggering. It's hard to get my mind around what 29 billion barrels of oil is, especially as we're trying to come up with alternatives for that, or 5 billion tons of coal. And you think of all the carbon from that, it's not surprising that we're adding a net of roughly 3.5 billion tons to the atmosphere each year. You can see from this the ocean is the biggest carbon sink, and only a tiny fraction actually ends up net in the atmosphere. So it wouldn't take a huge shift in what we capture in the ocean to see if we could reverse some of this, but obviously the best way to do it is to stop burning fossil fuels. We're trying to take some unique approaches to this. What I've told you up till now that started with our major sequencing efforts in the late 1980s through to today has been the digitization of biology. We're taking the analog world of biology through the genetic code, digitizing it, and putting it into a compute environment where it can be distributed easily around the world, analyzed, compared, and now we're going in the other direction. We're trying to take that digital information back, synthetically convert it back into an analog world to do new things. So I like to view these 10 million or so genes as the design components of the future. Many of you have made your careers in the electronics industry. In the 1950s, there was only a handful of components. There were resistors and transistors and capacitors. We have literally millions of design components to try and go to this next phase of human existence. So we've been trying to work on building a first synthetic microorganisms, and some of the reasons for this are as simple as basic science. Any genome that we sequence, 30 to 50% of the genes are of complete unknown function. Even this minimal genome that we've been working on, out of 300 to 400 genes, 100 are of complete unknown function. So we don't even know the complete component functionally of the most minimal cell. We started by making a virus a few years ago. Trying to synthesize DNA very accurately in large pieces is very, very difficult because it's a degenerative process, and we found ways to fix that. And so this is a case we went from the genetic code and the public databases for a bacteria virus, FiX174, to actually synthetically making that chromosome in about 2 weeks. We then took this synthetic chemical DNA and inserted it into bacteria, and the bacteria started making the virus. The virus killed the bacteria, and that creates these clear plaques on the plate. So this is a case where the software is building its own hardware. So we can take what the equivalent is of a computer program in the piece of a synthetic chemical DNA and put it in a cell or a cell system, and it starts building its hardware around it. We've been trying to now do this not with viruses, but with a bacteria, and we build viral-sized pieces of DNA, and we try and put them together to make an artificial chromosome. And one way to do this is called homologous recombination, where basically there's a number of enzyme systems that will assemble pieces of DNA. Now this happens naturally, and this is a surprise to many people. We have organisms that can withstand millions of rads of radiation. Don't try the experiment, but you can't. You'll be killed with a small fraction of that amount of radiation. What happens with these organisms, their genetic code gets blown apart into hundreds of little pieces, but you drop them in water within 12 to 24 hours, they reconstruct their chromosomes exactly as they were before, and they start replicating again. So those of you who think that the idea of panspermia, that organisms could travel through space and land in an environment such as Earth and start replicating, is not far out at all. These can take millions of rads of ionizing radiation, be completely desiccated, and survive that for extremely long periods of time. Here's actually what happens. The chromosome is blown apart into these tiny pieces, and the bottom panel shows that reconstruction after 24 hours. So we've been trying to isolate these mechanisms from the cell to create a robot that could manufacture 1 million chromosomes a day. And we're calling this new field combinatorial genomics. So imagine if we could make 1 million synthetic chromosomes every day, and then you can screen these for simple things. Do they result in a viable cell? Do they produce hydrogen? Basically, whatever we want to screen for becomes a possibility. And so this is a big change on what we can do with biology, where we're limited only by our imaginations. Compared to all that oil and coal, it's a small portion of energy compared to what hits the Earth constantly from the sun. We would only need a few square miles of the Arizona desert if we had 10% conversion efficiency to produce all the energy used on our planet. And these are our challenges. Biology can certainly do this, not at 10% efficiency, but we're trying to modify various systems to do that. So we've been modifying photosynthesis to take sunlight and convert it straight into hydrogen production. We've been doing this because we found in our ocean environments a new hydrogenase that's insensitive to oxygen. Oxygen actually kills the system. We found some rare molecules that are resistant to this. This is one way to go right from sunlight into producing energy. We're modifying another cell to break down cellulose into sugars and ferment those into unique alcohols, all in a single bacterial cell. This notion of energy crops is totally a new concept. And there's been basically little selection for what we are starting to define as energy crops. In this country, there's this massive push for converting corn sugar into ethanol, but I think it's already had negative side effects and is not going to be a viable approach for very long. In terms of just in the last 6 months or so, the cost of feed for farmers has almost doubled as people try to switch some of their crops into energy versus food. So we clearly need places and alternatives where people aren't making the choice between food or energy. Some things like switchgrass as a feedstock for cellulosic sugars are certainly one possibility, but we're looking at a number of others, and we're looking at synthetic microbes that can do very rapid processing of these into chemicals, food, or energy sources. Later this month, DuPont's opening a huge plant in Tennessee where they're using literally tons of modified E. coli to convert sugar into propane diol. Propane diol is a key component of their new Sorona polymer that they're making stain-resistant carpets and clothing from, and they argue that that's going to be the first truly multi-billion-dollar biotech product. We have things like palm plants that have as much as 10 to 20 times the output per hectare as other crops. Perhaps the most promising is these simple organisms from the ocean. People at Arizona State University have already modified some of these where they produce 50% of their weight as lipid. So imagine capturing this CO2 back from the environment. Sunlight is the energy source. They make huge amounts of lipid that if there can be effective ways to harvest this, can produce clearly things on the scale of what could replace oil. So here's annual production in terms of biodiesel. You can see palm oil certainly outranks everything else that is grown in this country, which is primarily on soybeans. But synthetic algae or algae with these modifications have tremendous potential when you think on the order of 20,000 gallons per acre. We're trying to design cells that can capture CO2, convert the carbon in CO2 into sugars, into proteins, into biopolymers, or into a variety of fuels. When you're not starting with oil, we're not limited chemically with what we can do. So the fuels that we currently burn are what can be produced from oil. We've been looking at a variety of fuels, and there's chemical compounds that we think we can make synthetically in bacteria that would be far better, safer, high combustion fuels that could be produced locally instead of in places where they have to be trucked or shipped around the world. So it gives us different capabilities that we've ever had before, and we're limited, I think, primarily right now by our imaginations. So future uses of these are certainly contribute to understanding of life. My goal is to replace the petrochemical industry in the order of the next decade, and hopefully a major source of energy, but there's not going to be any one solution that gets us off of oil and coal. But when you think about what can be done synthetically, we're looking at systems that can produce virtually an infinite variety of antibiotics and even antibodies in terms of vaccines. We've obviously been concerned with the ethical considerations of this, and if we're making new organisms that have never existed before. We stopped our research in the late 90s to undergo a year-and-a-half ethical review that we funded at the University of Pennsylvania. They published their results and suggested the reasons we were going and how we were proceeding were good. The Sloan Foundation has given us funding for the last year and a half, along with MIT and a Washington think tank, to look at laboratory standards for manufacturing new organisms and ways to do this safely. This was a big concern at the start of the era of molecular biology, putting human genes and bacteria, plant genes and bacteria, and with the literally millions of experiments that have been done with E. coli, which was engineered not to survive outside of the lab, this has not been an issue. My notion was that no human pathogens should be made. 2 out of 3 of the first synthetic viruses are human pathogens, one being the 1918 flu virus, but it's led to key understanding already, so of how to develop new vaccines against flu. This is the start of a new era, combined with other technologies. I think we'll have, I don't think it's an overestimation to say, mind-boggling impact on every aspect of our thinking about life, thinking about alternate sources of production, and some exciting ways to attract new young people into science. So it's a pleasure being with you tonight, and hopefully I've stimulated some of your thinking. Thank you. That's wonderful. Thank you.
Hi, my name is David Sakeley. I'm a private investor. You talked about panspermia. Can you imagine an experiment to try to collect possible DNA in Earth orbit or in outer space and detect whether there's really transit of genetic information between planets?
Yeah, and the answer is absolutely yes. The trouble is we've already grossly contaminated at least near space. I've jokingly said each time the shuttle went up or they flushed the commodes on the space station, billions of microorganisms got launched into orbit. I got into a little bit of trouble at actually a TED conference by saying that the outside of the space station was coated with feces and urine. And they've started now completely recycling the waste, and they don't launch it into outer space anymore. So I have no doubt that we'll find all kinds of Earth organisms in space, and that they can be sampled and collected there. I have no doubt we'll find organisms on Mars. We exchange roughly 200 kilograms of material between Earth and Mars each year, and that's a very near neighbor. If we don't find something there, I think most people will be extremely surprised. So we don't want to just confirm that we're contaminating space. We want to really see if the notion of panspermia could work on very long distances. For those of you who think about the 4 to 5 billion years of evolution on this planet, if panspermia is possible, it adds another 4 to 5 billion years to the time frame, which gives us some remarkable time for great diversity. So I think we're limited right now by sampling techniques. We've been working on designing some filtering systems that could capture things, at least in near orbit. Obviously, we'd like to get samples back from Mars, but none of that will prove whether it's panspermia just by finding similar organisms in both places. But I think it'll be a challenge for getting out our reach a little bit further.
Yes, Ty Carlson with Microsoft. Have you done sampling of the seawater, for example, 100 feet down or something, where all of a sudden you don't have photosynthesis, where to see any commonalities between that, where light isn't necessarily present and these organisms aren't sensitive necessarily to the light within the first 30 feet or so?
Yeah, I mean, the initial expedition has been mostly just surface waters. We have done some deeper sampling, but not enough to get totally out of the realm of light. So we're trying some samples from deep ocean vents and other areas where some of our organisms have come from, but they were, biological isolates versus trying to do everything that's down there. So, when you look at the diversity that we've shown, we're literally just talking about the upper few meters. It's not even the maximum density. So chlorophyll density is roughly around 60 feet below the surface. So there's more photosynthesis 60 feet down than there is right on the surface. So I think for the next decade or so, we're going to see the documentation of diversity just growing and growing. How far we can go down, I'm hoping we'll find photoreceptors that can deal in the ultraviolet. When you imagine trying to get increased efficiency from the sun, if we take all these biological photoreceptors, which transmit electrons across a membrane, and we could deal with the entire spectrum of light, we could make some pretty efficient solar collectors. So we're anxious to see what else is down there. I don't know if we're limited to the microphone or if people want to just shout. But
Reese Jones from Berkeley. What do you think about patents of genes?
Well, it seems to be a question I'm getting asked a lot tonight. It's an experiment that was tried on the bulk scale when we developed these methods to isolate them. The U.S. government tried to patent all the discoveries that I made. I think the only winners in the large-scale patenting of genes have been patent attorneys. And I seem to be very popular with them because I've made so many discoveries. But there's places where patents with things that have direct clinical implications or applications are very important. People who are dependent on insulin are probably pretty happy that Genentech and Eli Lilly have a patent and can produce human insulin. So on the specific cases, it's extremely important and valuable. On a bulk case, a lot of governments wanted to tie up and own the data that we discovered in their waters and didn't want it to be public. It's actually gone a complete circle from what people thought was bad, what various people were doing with the human gene discovery, and where there was potential for direct therapeutic benefit, to now with this massive amount of environmental data that is in many cases being restricted either from sampling or from data release. One good thing about patenting, it forces release of data. If countries want to spend their money on patenting as long as the data is available, I don't really care, because the patents aren't going to do them any good. So hopefully that experiment's been done and nobody will waste much money on it in the future.
Greg? Stuart Brand.
Yeah, Stuart.
You made mention of the large number of microbes that are resident in and on humans, like probably our human cells are 1/10 of the cells that we carry around. How about doing an inventory of all of those creatures?
Well, I'm not sure an inventory helps so much. I mean, there's a lot of variation. So we're continuing to do more metagenomic studies. So the Dental Institute has funded us to look at the oral cavity, and what a number of military dentists have observed is the flora in our mouth changes totally based on what environment we're in. So soldiers coming back from the desert have a totally different set of microbial flora than people living in New York City. In fact, they are predicting that you can tell where somebody's been living for a while by all the bacteria that reside in their mouths. We have a project called the V Project, so it's looking at the vagina. We've published the first paper on the intestinal flora. We're breathing in, as I indicated, huge numbers of microbes and viruses on a continuing basis. We have a large number of organisms resident in our lungs and airways. And if you take antibiotics, you're totally turning over a certain part of your collection and trying to pick up a new one from somewhere else. H. pylori, the organism associated with ulcers and even stomach cancer, by tracking the genetics of that organism, you can work out more in the way of genetic linkages than from sequencing the genome. So it's exchanged early on with family members, and you can track whole lineages by looking at just that bacteria. So it's a large repertoire of organisms that changes pretty dynamically. Trying to understand that and its association with human disease, I think, is a very worthwhile goal. But we're, as Mark said, we're definitely not alone. Just because it's just not part of our normal sensing apparatus. There's a couple of questions down here in the front. I don't know where the microphones went.
Okay, Eric Lang, Microsoft. The precautions around human genomics was interesting. Is there also a danger as we turn toward environmental sciences and energy that there is an equal danger in introducing new invasive organisms or pathogenic organisms into the environment or the food supply?
I mean, if there's going to be unscrupulous scientists and companies, the answer is yes. But I think that's a fairly low risk. It's interesting. I talked to some people in Homeland Security when they first learned of this, and in their scenario, which I think they watched too much television, was that pharmaceutical companies were going to develop new killer organisms that they would have the only cure for, so they could make a fortune by introducing infectious agents and then making money by having the cure for that. I think no matter what you think of pharmaceutical companies, I think that's a pretty unlikely scenario. I think trying to design an organism that could robustly survive in the environment is going to be a huge challenge. It's much easier to design one that won't. and to design one that won't live outside of the laboratory. And that's part of the guidelines that we put together. But it's very recent history where the U.S. government and the former Soviet Union government were spending vast millions on trying to get new killer varieties of microorganisms, for warfare purposes. So if that kind of thinking exists, then yes, it's a danger, but we're more of a danger to ourselves from trying to make deliberate killer organisms than from trying to find something that produces an alternative to oil.
Hi, Greg Ellis from OnStream. A brief question, kind of a follow-up on that one. What is the threat level, in your opinion, on genetic weaponry in the future?
I'm not sure I'm in a position to ascertain what other people might do with this technology. We're told what a huge threat bioterrorism is, but more people get killed by lightning bolts in the U.S. alone each year than by the whole history of bioterrorism events. Are they hypothetical possibilities, and are there lots of people that want to do harm out there? Yes. Anybody that wants to get anthrax can go to any farm in the world, take a soil sample and have anthrax. So I don't think you need to go through the complexity of trying to design an organism if you want to do somebody harm. That being said, any virus that exists that has been sequenced today can be manufactured. But a lot of viruses, the DNA itself is not infective. And so you can't just boot up any life form just from having a piece of DNA. It's certainly a concern. It's a concern of our government. And I think a key feature of this is I make zero distinction between a man-made microorganism and a new emerging infection. As I said, from a single virus, from HIV, numbers have been revised on the order of 3 billion people. That's a pretty big impact from an emerging infection. We're seeing other examples of things. I think we need to come up with antimicrobials, antivirals that can protect society across the board, whether it's an emerging infection or a man-made infectious organism. It won't have any impact on the treatment by knowing which it is. We need new approaches to making these, and I think this technology provides that.
Yeah, Craig, actually following up on that point, it's Jeff Nick from EMC.
Yeah.
I was wondering if all of the collateral and inventory that's been created is actually being turned into targeting the variance between organisms at the viral or bacterial level that might suggest better ways of dealing with infections and other organisms that are damaging by different gradients of environmental change, for example.
Well, what it does, it gives us an indication of when we're looking at infections that we're probably not looking at a single clonal organism. We're looking at a broad spectrum of related organisms. I think most people are unaware of organisms like Haemophilus influenzae that lives in everybody's airways unless they're on massive IV antibiotics. Basically renews its genome every 20 minutes or so. And if we develop antibodies to it foils our immune system by constantly changing its cell surface proteins by some pretty simple mechanisms and variation of copying its own genetic code. So we will never, as the Surgeon General said in 1969, win the war against microbes, You know, we're not even staying even right now with drug resistance and all the variations that occur. So we need to develop a new set of tools either way. But these new tools, you know, I only showed you a fraction of what we're working on. We can now sequence an entire genome from a single bacterial cell. So we're trying to look at mechanisms, for example, in blood infections, where current technologies, you wait 3 or 4 days for an answer, what's the infectious agent, to where from a blood sample we could decode an entire genome in a few hours, or look at the entire spectrum using these same approaches of what's in the blood to try and understand what an infectious agent is and what the course of things are. So, you know, these are new tools taking us into worlds we've only wondered about before. And I think that that's an area that I would hope would be followed up on.
Thank you, Craig.
Thank you.
Brilliant. Thank you, Bill. Thank you very much.